Picture this. You're sitting at your study desk, looking down at a densely packed biology textbook page and wondering how on earth to make sense of all these rules of nature. Well, don't worry, because today we are demystifying biology in this explainer. We're going to break down the exact logic behind how scientists classify life and then walk step by step through the scientific method. We'll turn all those overwhelming concepts into clear, actionable knowledge you can actually use.
Our guide today is a foundational textbook page covering classification and the scientific method. We're stripping away all the extra noise and going right back to the core principles. It's laid out perfectly, kind of like a classic Q&A, giving us that fantastic structured classroom vibe. We're going to extract the core wisdom directly from these paragraphs so you can truly master the why and the how of biological research.
Here is our exact roadmap for today. First, why classify biology? Second, the power of specialization. Third, the scientific method. And fourth, step-by-step experimentation.
All right, let's jump straight into Section 1. Why classify biology? Organizing life on Earth.
Think about the sheer scale of life on Earth for a second. I mean, from towering redwood trees to your family dog, all the way down to the microorganisms floating everywhere around us right now, it's absolutely massive. To manage this vastness, scientists have to use classification. By dividing biology into dedicated branches like botany for plants, zoology for animals, and microbiology for tiny organisms, researchers can really focus in without getting overwhelmed. By not trying to study literally everything all at once, their research becomes infinitely more effective.
Let's put that into perspective. There are more bacteria in your mouth right now than there are people on Earth. Microbiology focuses exclusively on those tiny, microscopic organisms. If biologists didn't specialize like this, it's highly unlikely we'd ever have developed the tools and intense focus needed to understand a world we can't even see with the naked eye. Narrowing the focus actually expands the depth of our knowledge.
Moving on to Section 2, the power of specialization. Let's talk real-world impact. So why does all this matter outside the classroom? Well, because specialization leads to massive real-world impact. Botanists develop incredibly tough crops designed to survive harsh droughts. Zoologists figure out the absolute best ways to protect endangered wildlife. And microbiologists, they discover new life-saving medicines. Plus, by becoming absolute experts in these smaller branches, they can team up across fields to tackle massive global issues like climate change or widespread diseases. Specialization literally makes life better for everyone.
But that brings up a really important question. How do these highly specialized scientists actually prove their observations are true? How do we move from just a simple guess to a verified scientific fact? Imagine you're a researcher looking out at a forest and you notice something strange happening. How do you definitively prove that what you're seeing is an actual rule of nature?
That brings us to Section 3, the scientific method, taking it from guess to fact.
The scientific method is the universal tool researchers use to turn a mere observation into confirmed, reliable knowledge. It follows 4 crucial steps: hypothesis, experimentation, data collection and analysis, and finally, conclusion. This is the gold standard, guys. It doesn't matter if you're studying giant elephants or microscopic bacteria. If you want to prove something, you absolutely have to walk this exact, repeatable roadmap.
Let's see this in action in Section 4. Step-by-step experimentation, testing plant growth.
Let's take that theoretical roadmap we just talked about and apply it to a real scenario, straight from your textbook. Say you notice a certain plant thriving in the shadows. Step 1 is turning that observation into a hypothesis. You write down, the plant species grows more quickly in shady places than in direct sunlight. Now remember, it's not a fact yet. It's simply a proposed explanation that we now have to put to the test.
To test this fairly, a scientist sets up 2 distinct groups of the exact same plant species. On one side, we have the experimental group. These are the plants placed in the shade, which is the specific condition we're investigating. On the other side is the control group, plants placed in direct sunlight. This control group is absolutely vital. It gives us a baseline to compare against, so we can ensure we're isolating the specific effect of the sunlight and literally nothing else.
Now, for this to actually work, everything else in the environment must remain identical. We call these constant variables. Factors like the soil type, the amount of water, and the nutrients provided all have to be strictly constant for both groups. Think about it. If you gave the shady plants extra water, you wouldn't know if they grew faster because of the shade or because of the extra drink. Controlling these constant variables is the absolute heart of a valid experiment.
Next up is the moment of truth. Step 3. Data collection and analysis. You don't just look at the plants and guess. You measure specific, objective metrics. Over a set period, you might measure the physical height with a ruler or precisely count the exact number of leaves in both groups. You gather the hard numbers so you can directly compare the real results between the sun and the shade.
Finally, step 4, the conclusion. This is where the data makes the final decision for you. If your measurements show the shady plants did indeed grow faster, your hypothesis is confirmed. But if they didn't, it's time to rethink the idea. And honestly, here's the best secret of science. A rejected hypothesis isn't a failure. Not at all. It's a highly successful step in learning what doesn't work, and it guides you directly to a much better question. In science, being wrong is just another way to win.
Science is just a continuous loop of asking questions. So let me leave you with this. If your experiment proved that shade didn't help the plant grow faster, what new variable would you test next? Soil temperature? Water levels? You see, the scientific method isn't just for textbooks. It gives you the ultimate tools to investigate absolutely anything you're curious about. Think about that the next time you step outside. Take out your notebook and observe the fascinating world around you.